right, so then our next team up is Toros Neuro team. Come on up.
Welcome everyone. Today we're excited to present our cutting-edge innovation that gives a glimpse to a future treatment of Huntington's disease. We are Tetrahedral Toros with our product Neotetra. I'm Naomi Ordóñez.
I'm Lizette Carrasco.
I'm Melissa Mershane.
And I'm Brian Serrano.
So the problem we are tackling is Huntington's disease. And what is Huntington's disease? It is a genetic neurological disease where neurons in the brain break down over time. And in this image, you can see the mutated HTT gene that then leads to the CAG repeats. This disease causes a combination of severe physical, cognitive, and psychiatric issues. There's currently no cure for Huntington's disease and affects 1 in every 10,000 people in the US and is increasing 15 to 20% per decade. It typically occurs in people's ages 30 to 50, but becomes progressively fatal over 10 to 25 years.
Current treatments for Huntington's disease treat specific symptoms such as chorea, depression, psychosis, and mood agitation. Although this is the standard of care today, it is ultimately not a cure. As the disease progresses to later stages, the cost of medication becomes more expensive. Current clinical trials are AMT-130, which uses an adeno-associated virus vector to deliver microRNA into the striatum. ASO-tominersen is an RNA-targeted therapy distributed throughout the central nervous system. HNSC-01 is a stem cell therapy to protect existing neurons. However, these procedures are highly invasive, requiring surgery or lumbar puncture, leading to risk to patients, in addition to the neuroimaging and post-operative care, which lead to high medical costs.
Our product is a DNA tetrahedral nanostructure that delivers microRNA to neurons in the brain. But why a DNA tetrahedral nanostructure? It's similar to DNA origami, and it has a 3D rigid structure that allows for stability and protects our microRNA from degradation. It also improves cellular uptake and has natural properties that allow it to enter the brain, such as its biocompatibility, its small size, and its programmable structure. But how do the microRNAs attach? So, the microRNA and DNA strands are complementary to each other, which allow them to directly attach to each other. And it also has regions that allow the microRNA to be cleaved off and delivered. And we plan to deliver it through the nose-to-brain pathway, which allows us to directly avoid the blood-brain barrier. Next slide. We plan to use the RVG peptide, which is rabies-derived, and it allows for brain delivery to be improved along with neuronal uptake, also increasing targeting efficiency to the striatum, specifically to medium spiny neurons, which are among the most effective neurons in Huntington's disease. Our approach is better because the nose-to-brain pathway allows for our approach to be non-invasive in comparison to the other approaches that were mentioned in the past, like AMT-130. And our DNA tetrahedral nanostructure is stable and efficient for microRNA delivery. We aim for early-stage patient outcare, ultimately providing individuals with affordable care.
Yeah, developmental process in Milestones is focused on 3 critical stages. In stage 1, our ultimate goal is to see if our product is safe and effective, which is why we're asking for $6 million in seed funding to provide proof of our concept, making sure it is targeting where it is supposed to. This will then lead us to begin hands-on experiments on mouse studies to continue testing the effectiveness of our product. Then in stage 2, we seek $10 million in Series A funding that will help us begin preclinical research specifically on different animal models. Establishing our ultimate goal being to establish the overall safety, tolerability, dosage, and efficiency of our product. Now in stage 3, we seek $40 million in Series B funding to begin phase 1 of our clinical trials. All 3 stages coming together to sell our platform to a larger pharmaceutical biotech company. So now, where does our profit come in? We would be acquiring profit from 3 different sources. 1, from selling our platform for $200 million. Another, salary if we continue with the pharmaceutical company. And lastly, profit from our 4% royalty rate.
Overall, we are building a programmable brain delivery platform for neurological disorders other than Huntington's. We want to be able to deliver treatments safely and effectively to the brain with our RNA therapeutics, non-invasive intranasal delivery, and targeted nanocarriers. Our vision is to create a scalable technology that could transform the future of precision medicine for neurological diseases. Thank you.
Excellent presentation, really nice props. So tell us about how clinical trials work. How long does it take? How much does it cost?
So for the clinical trials, we'd be hoping it'd be like around $200 million. Yeah, so it'd be around two. So the clinical trials would usually take around.
So those are some estimates for the funding with clinical trials, but requiring the time, we are estimating 7 years. But it would depend, first of all, on the first clinical trial, such as, if there is any difficulties or something, then that could increase the time. But if it is all going how planned, it should take up to 7 years to reach our ultimate goal of selling to a pharmaceutical biotech company.
In your animal models, which animals do you envision testing this on that would mimic humans?
It would start with mouse studies, as it says in stage 1. I think that is just our ultimate goal, to see how it goes in mice studies, most likely.
And let's say it was successful in mice, then what do you do next?
It would go to, so once we verify the safety of the tolerability, dosage, and efficiency with the mouse studies, that's when we move to being able to just sell it to a larger pharmaceutical biotech company so they could continue building the clinical trials.
Very good, thank you.
You have preclinical that says different animal models. So what animals would it be there?
So in addition, just like as we see right here, there has been other animal models such as sheep studies that have been using bioavailable components as well. So we would probably be using sheep as well, in addition to my studies.
This is a very interesting topic, and I'm very impressed that you would tackle it. So how did you get drawn to this? Does somebody have Huntington's or Alzheimer's or neurological disease in a family member? Or?
So in my family, my mother actually passed away from Huntington's disease, which especially motivates my interest in neuroscience and medicine. And we all are also neuroscience interested. So that's what especially motivated this topic.
Thank you. One more question. Did you guys 3D print your prop? Who did that?
Yes, it was done at our school. We're from a STEAM academy, so we have a 3D printer with our engineering teacher, so he was able to help us create that.
Very cool. Thank you.
Hi guys, awesome presentation. Yeah, I have another question on this, milestone slide. I guess, I really like, number one, I want to establish that I really like that you guys established, different funding, goals at different actual clear milestones and why. But I mean, as is the case with a lot of these companies where, you have these established milestones, inevitably some things overrun, right? And milestones, they almost never actually get hit on time. And so, and that's an understatement, honestly. And I guess what would be the theoretical big tangible proof of concept, I suppose, between theoretical stage 1 and stage 2 funding, right? Like, what is the most important thing for you guys to establish to actually get that second round?
I think what's most important is obviously to make sure that we're able to carry this safely with the dosage and everything that we're putting together, we want to make sure it is safe, which is why we're starting with the mouse studies. So I think that's the most important part, is just ensuring its safety and everything so that we could overall be able to ultimately sell it. So safety is our number one priority, because we don't want it to be like other treatments that are currently available that can be invasive and just not overall the best safety.
On your first slide, you don't need to go back to it. You mentioned 3 different kind of branding things. You mentioned, sorry, could you hold this a second? I need an octopus. You mentioned, yeah. So you mentioned on the first slide, and I apologize if I mangle the words, MicroRNA, Neotetra, and then Toros Neuro. Pick one. Pick one. Yeah, you know, pick it. And there's just something I need to say. What is your name, Brown?
Alyssa.
Alyssa. I lost my mother to breast cancer when I was 16. And I thought about going into science because I thought I could help cure breast cancer. I want to tell you, don't stop. You may find another path. I found the other path to work in a place like this that works on cancer therapeutics, vaccines, that sort of thing. So don't stop. Go where your heart leads you, okay?
All right, thank you very much.